Okay, good afternoon, everyone. Today I'm just going to only be talking very, very briefly. This is because, first of all, we ran way over time on one of our classes this week, which I hope you've still completed. If you haven't, please make sure that you do. The section on serology is really important and segues into this next section where we're going to be talking about what is probably one of the most important aspects of forensic biology, and that is the use of DNA as evidence, how DNA is used as forensic evidence. I've said several times in this class, and you watched a movie about the doubts that lie around things like pattern analysis in forensic science, because we have to put air quotes around that word science when we talk about several of these practices. The reason that they are not science is that they are not data-based, and they are not very often not based on experiment or on tested hypothesis. They are based, many of them, purely on observation and often upon subjective interpretation of what is observed. Scientists look at that and say that that is not true science. But DNA analysis is truly scientific. It is truly scientific because it is based upon experiment. And the point about experiment is it allows us to set statistical boundaries. It allows us to say, we believe this to be true. We believe it is true within these particular boundaries. We end up taking DNA and we present the DNA, and we can say the likelihood that two people have the same DNA is 1 in X billion. We can't take a fingerprint and look at it and say the likelihood that another person has this fingerprint is this number, because that has never been established. You can't establish it by experiment or by true scientific observation. So DNA is tremendously important because it is based on such solid science, and it has revolutionized, as you know, it has revolutionized forensic investigation. Of course, the great disadvantage is DNA is not always present. A perpetrator does not always leave DNA behind as a trace of their presence at a crime scene. Nonetheless, it does happen. And it happens with remarkable frequency. And when a biological sample is recovered at a crime scene, which may be linked to a perpetrator, the analysis of any DNA present becomes one of the most important pieces of forensic evidence that can be gathered. So we need to understand how this is done. But I also want to use this as an opportunity to tell you about DNA itself and about its structure and its functioning a little bit more broadly than is covered in your text. So I'm going to go through this relatively slowly. And today we're just going to have a short introduction, and then there is a movie for you to watch, which is essential viewing. You must please now, today, sit down and view that just as if I was presenting it to you in class, because you're going to be quite lost if you don't watch this movie, because I'm going to go on the next week to discuss actual DNA structure before we talk about its function and how it's used in forensic biology. Okay, so the very first thing that we've got to get clear in our minds, DNA, we hear it the whole time. That is an acronym for its chemical name, which is deoxyribonucleic acid. You will understand each part of this, the deoxy part, the ribo part, and the nucleic acid part by the time we are finished talking about structure and everything of DNA. So that's its full name, deoxyribonucleic acid. So the very first thing we need to understand is what a nucleic acid is. Well, there are several nucleic acids that are used in our cells. And many of them coordinate with one another. And they coordinate with one another for one purpose. And that is to create proteins, largely proteins, in our cells, which are required for the running of our cells. Nucleic acids are polymers. That means they are made up of small units, which are all joined together. The cell joins them together in specific order. But there's something really important to realize. The nucleic acids use only 4 monomers. The small units are called monomers, and the monomers are joined together to form the polymer. Nucleic acids use only 4 monomers. But here's the thing, they each have their individual identity, but you can join them together. They could be joined together in any order. So the order, the number of orders that they could be joined in is infinite. The cell joins them in a very specific order. And that order of nucleic acids actually means something. Now, the molecule that we are mostly going to concern ourselves with is DNA. And DNA is a nucleic acid. There's another nucleic acid we're going to encounter called RNA. For the moment, let's just concentrate on the DNA. Because DNA is a recipe. It is a recipe for all of the proteins in your body. And the recipe is written in words that are composed of letters. And the letters are the 4 bases or monomers that get joined together to form the polymer, which is DNA. So there is, to make a protein, we need a particular long polymer with these bases in order, spelling out the constituents of the protein. That long section of DNA, which spells out the recipe for a protein, is a gene. That is what a gene is. It is a recipe for a specific protein. In general, for our purposes, we can say for each one gene is one protein. And that is the nucleic acid that is going to be our main concern, is DNA, deoxyribonucleic acid. And the deoxyribonucleic acid specifies the structure of a protein by the sequence of monomers. Each monomer is called a nucleotide. You will also hear it referred to as a base for reasons that you'll realize in a while. But each monomer is a specific chemical unit called a nucleotide. And in DNA, there are 4 nucleotides, 4 distinct nucleotides, different structures, but they can be joined together in any order. So the number of proteins that DNA can code for is enormous, absolutely enormous. But each protein has its own specific recipe. Now, that's not the only thing. When we're talking about forensic science, we also need to realize that DNA there's a huge amount of DNA in our cells. If we were to spin it all out, it would be meters long from each cell. The genes only occupy a certain amount of the space on our DNA, and in between the genes, there are long areas which do not code for proteins, but they contain very useful information. And when we sequence DNA in order to identify an individual, it's very often those segments of DNA which intervene between the genes, which are of particular interest, as we'll hear later. But again, there's nothing to distinguish them from the rest of the DNA. They're still composed of nucleotides joined together in a specific order. Okay, so the structure, first of all, let me tell you that we've known about DNA for a very long time, since the 19th century. In the 19th century, there was a lot of interest in the nucleus of the cell, because it was observed that during cell division, the nucleus underwent some spectacular changes as a cell divided. It formed chromosomes, and there came to be an association in people's minds between the chromosomes that they saw in the nucleus of the cell and heredity. There were certain things about the chromosomes which suggested that inheritance had something to do with the chromosomes. There was a lot of interest in the chromosomes. How is it possible that they were conveying hereditary information? It wasn't certain, but they made an association, and they experimented to find out what is the structure of the chromosomes. And they found that the chromosomes were made of two things. The first thing that structures the chromosomes is nucleic acids. They were called nucleic acids because they extracted them from a nucleus, and they are very weak acids. They're biological acids, very, very weak acids. But the acids they are, so they call them nucleic acids. And they also saw that in the nucleus, there was a lot of protein. Now, proteins of nucleic acids on the face of it are relatively simple because they're composed of these 4 nucleotides all joined together into very long sequences. But it's very simple, actually a rather simple molecule. But the proteins that were in the nucleus are very, very complex. And there's many, many, many of them. And they have very different structures and very different chemical structures and behaviors and everything else. So everyone was convinced that if the chromosomes in the nucleus were conveying hereditary information, which is very complicated, it was probably the proteins that did it. And there was a huge argument because there was a group of people who said, no, the DNA, this nucleic acid is the hereditary material, but we don't know how or why, because nothing was actually understood about the real structure of the DNA. The other group said, no, it has to be the proteins because the proteins are very, very complicated. So they must be conveying all of this information. It took a long time, it took nearly 50 years to prove that, in fact, it was the DNA that was the hereditary material. We're not going to discuss all the reasons why that decision was made, but it was a huge fight amongst chemists and biochemists and biologists as to whether it was DNA that had the hereditary material or the protein. So they used to throw things at one another at conferences and everything. It got very, very heated and friendships broke up over the whole question until it was eventually settled. DNA is the hereditary material. Once that was decided, everybody started to try to find out what was the structure of DNA. What was the structure of DNA that allowed it to convey such a huge amount of information? They knew by that stage that there were these 4 nucleotides: adenine, guanine, cytosine, and thymine. Remember those names. You're going to hear them over and over again, and you need to remember them: adenine, guanine, cytosine, and thymine. Those are the 4 nucleotides that make up DNA. But nobody knew how is it possible, just those 4, that DNA can be holding so much information, all of the information to run the complexities of a cell. The first thing that needed to be found out was what is the structure of DNA. And this was a fascinating struggle, a scientific struggle all on its own. And this is what I want you to do once I've finished talking, is immediately watch this movie, please. It's from the Howard Hughes Medical Institute, and it discusses the work of these 4 workers: Rosalind Franklin, Harold Wilkins, James Watson, and Francis Crick. Some of those names you may well have heard, because the two that everybody remembers are Watson and Crick, largely because they were relentlessly self-promoting. But that's another story. In any case, all four of them did win the Nobel Prize for this work. I'm sorry, three of them won the Nobel Prize for this work: Crick, Watson, and Harold Wilkins. Unfortunately, Rosalind Franklin had died by the time the Nobel Prize was awarded, and it's never awarded posthumously. So these three gentlemen, they won the Nobel Prizes, and it was probably one of the most important scientific findings in human history, was the structure of DNA, because just by explaining the structure of DNA, they were very simply and elegantly able to show how DNA codes for protein, what the structure of a gene is, and simultaneously how it is that when a cell divides, it can hand over that information to its daughter cells, which was obviously critically important. Every cell needs to inherit all of the necessary information. And their model for the structure of DNA explained that very simply, very elegantly. Okay, so I'm going to stop there, and I want you to please now sit and watch this movie. All right, and I'll see you on Monday, and discuss on Monday. We will discuss the actual structure of DNA. We will repeat some of the information which is in this movie.